Types of centrifuges and rotors. Myself, Jumana Karadan, 2nd MSc Biology from St. Mary's College, Thrissur, in the guide of Dr. Kain Vadakan, Department of Biology. Introduction. Centrifugation is the separation technique commonly used in clinical and research laboratories. It is based on the behavior of particles in an applied centrifugal field. That is, more dense components of mixture move away from the axis of centrifuge, while less dense components of mixture move towards the axis. Types of centrifuge. Basic centrifuge consists of two components. One is electric motor with a drive shaft to spin the samples and a rotor to hold the tubes or other containers of the sample. A wide variety of centrifuges are available, ranging from a low-speed centrifuge used for routing pelleting of relatively heavy particles. There are three types of centrifuge. First one is low-speed centrifuge. Most laboratories have a standard low-speed centrifuge used for routing sedimentation of heavy particles. Low-speed centrifuge have a maximum about 4000 to 5000 rpm. Operate at room temperature with no means of temperature control. Two types: fixed angle and swinging bucket. It is used for the sedimentation of red blood cells until the particles are tightly packed into a pellet and supernatant is separated by decantation. Second one is high-speed centrifuge. It is used in more sophisticated biochemical application. Higher speeds and temperature control of rotor chamber are essential. Maximum 50,000 to 20,000 rpm. Operator can control the speed and temperature which is required for sensitive biological samples. Three types of rotors available here: fixed angle, swinging bucket, vertical rotors. And the third one is ultracentrifuges. It is the most sophisticated instrument. 65,000 rpm. Intense heat generated due to the high speed. Thus, spinning chambers must be refrigerated and kept at a high vacuum. Used for both preparative work and analytical work. Then, centrifuge rotors. Rotors in centrifuge are the motor devices that house the tubes with the samples. Centrifuge rotors are designed to generate rotation speed that can bring about a separation on of components in a sample. There are three main types of rotors used in centrifuge, which are fixed angle rotors. These rotors hold the sample tubes in an angle of 45 degrees in relation to the axis of rotors. The sample tubes at an angle of 45 degree in relation to the axis of a rotor. In this type of rotor, the particles strike the opposite side of the tube, where the particles finally slide down and are collected at the bottom. Fastest than other type of rotors as path length of the tubes increases. Second one, swinging bucket rotors or horizontal rotors. Swinging bucket rotors hold the tubes at an angle of 90 degree as the rotor swing as the process is started. In this rotor, the tubes are suspended in rags that allow the tubes to be moved enough to acquire horizontal position. In this type of rotors, particles present a long direction or path of force that allows the particles to be moved away from the rotors towards the bottom of the tube. Because the tubes remain horizontal, the supernatant remains as flat surface, allowing deposited particles to be separated from supernatant. Third one is vertical rotors. Vertical rotors provide the shortest path length, fastest runtime, and highest resolution if all rotors. Here, tubes are vertical during operation of centrifuge. Yield of rotor is not as ideal as position of tube, does not align with the direction of centrifugal force. As a result, instead of settling down, particles tend to spread towards the outer wall of the tube. These are commonly used in isopycnic and density gradient centrifugation. So thank you.